Thank you so much, Sanya. This is really a treat to be talking to the Bio-Ontologies and the ISCB community here today about our work on machine learning and ontologies and using these together. So I'll jump right in. So the background and kind of framing for what I'll be talking about today is the observation that will not be news to anybody in the Bio-Ontologies community, that what we kind of discover feeds into our open knowledge bases and our ontologies and so on. But then also from what we have discovered and this amassed and assembled volume of knowledge, this kind of feeds back into how we interpret and analyze our new results. So there are synergies there between kind of knowledge discovery or learning new things and knowledge capture and assembly and curation, where we want to build our databases, build our representations about what we know and integrate everything and make it all fair and discoverable and so on. So on the one hand here, we have this kind of traditional view of the scientific method, which is all about generating data and interpreting data based on a hypothesis and then coming to some conclusion. And the traditional view of the scientific method, I think, it leaves out this aspect that we work on in the Bio-Ontologies community or in the broader computational biology community of taking what has been discovered and putting so that those knowledge bases become more complete. And then in the end, we can use that knowledge and really make discoveries with the knowledge. So there's an extra bit here, which is about open data and open knowledge. And this extra bit is somehow, in my mind, kind of central to this knowledge discovery process or the making new discoveries. And I think this is a really interesting evolution of the scientific method towards a data-driven scientific method. So this is the background and the framing of the different approaches that I've been working on. But now I will get more to the kind of domain-specific topic and what we actually do. So as a background, we work on metabolism, on metabolic data. Small molecules are that part of biological processes which we somehow our environment. So what we do in the body, they are the carriers of the body and so on. So basically everything that's not proteins or encoded directly by the genome, this part of biological processes is small molecule metabolism, and it's really super important for lots and lots and lots of different things which we're very interested in at the moment, especially sort of trying to improve health over the lifespan, make people live in a healthier way for longer and so on. Metabolism is at the core of this part of the biological picture. And in the space or the domain of where metabolism is described, of course, there are lots and lots of resources which describe metabolic pathways as a whole, like which molecules react with which molecules and have which effect. But in order to disambiguate the small molecules themselves, you have resources which describe and classify small molecules. So these resources typically have at their sort of center of currency a structural representation of the small molecule. So here you have the molecular structure of caffeine. Caffeine is my favorite drug, maybe other people's favorite drugs as well. And this resource called KEBI, maybe some of you have heard, it's the largest publicly available ontology of small molecule metabolites that is available in the community. And I used to work for KEBI for a while, so I know it quite well. And it's a really great resource, big ontology. It has more than 100,000 molecules in it. But yeah, so this resource is classifying molecules, and what that means in practice is that there's a large sort of ontology of classes of molecules, and for the most part, these classes of molecules are based on features of the chemical structures, but there is also a separate classification based on what the molecules do, which kind of activities they can have in living systems, and also which organisms they have been found in as metabolites. So there's a lot of rich information stored in this ontology. And as I mentioned, the information is kind of clustered around the chemical structure. This is a visual representation. You have also other representations of the chemical structure. One really important one is called SMILES. That's a cute name. It stands for something like molecular line representation. I can't remember what the S is for, maybe system. But anyway, it's what's called a line representation of chemical structures. And that means that the atoms, so carbon atoms, are placed next to each other if they are bonded together. So here a carbon, a nitrogen, some more carbons. There's a branch. Branches are shown within brackets. So that would be one of these two branches here. Do you get the idea? The SMILES is another representation of the same information that we have here in the visual representation of the chemical structure. And there's other metadata such as the mass of the molecule, what its name is, and so on. And in the ontology, you have other kinds of relationships. You have classification, which is shown here as black arrows, but you also have, for example, has part. So we will not make a huge amount of use of all the rich complexity of information that's available in CheBI for what I want to tell you about today. Mostly we're using the classification hierarchy, but all the rest of the information is also really potentially usable, and that's really great. So the challenge that motivated my starting to work with machine learning in this particular area was the observation that the manual part of maintaining a large bio-ontology such as CheBI is limited and slow. And on the other hand, it's really essential because it's this manual checking by experienced chemists that makes sure that what goes into an ontology such as CheBI is so great and such high quality. But on the other hand, it's slow and it doesn't scale quickly enough to address the need. So during the time when I was working on CheBI, we would regularly get requests for kind of large scale collections of molecules to be added to CheBI, which would take longer than we had available in terms of manpower hours in order to them. So this was really a limiting factor. What you see here is the growth in terms of fully annotated compounds in CheBI over time, ever since its beginning. It's more than 10 years now since it started. This axis here is in releases rather than dates, but the releases are roughly monthly, so that gives you an idea of how long it's been going. And the gradient of this plot is more or less linear in the number of curators. So here you can see slightly steeper gradient, which corresponded to having extra manpower, and here you see a slightly shallower gradient, which corresponds to having a smaller curator team. And that's basically linear in the number of curators, and that will clearly never scale to the size of chemical space of what's available, right? So of what's needed in a way, if you want to interpret all of biologically relevant chemistry. I mean, so this illustration here shows you the size of different resources of small molecules. And here's kept, and it's in a log scale to fit everything in without completely dwarfing the CheBI. So here's our fully annotated molecules or classes in CheBI, which is over 50,000. I think it's over 60,000. And then you have PubChem, which is a large-scale publicly available resource of chemical data, which is, it's not an ontology, it's a database, but it has 100 million chemicals in it. It's a deposition database. I think many of the molecules in there are biologically relevant, not all. And then you have, let's say, other resources which are calculating all possible molecules up to a certain size. This is a hypothetical molecules up to the size of 13 atoms, and this is 500 million. And of course, you get much bigger collections of hypothetical molecules. But I think this very, very large scale is not what we need for biologically relevant chemistry. What we need for biologically relevant chemistry is, however, much larger than what we have in ChEBI, and much larger than what the manual maintenance can scale to. So it seems clear that there's a need for automation in this growth problem of growing the ontology, but it's also really important that the automation doesn't drown out the good quality of the manually annotated core. So when I was still working with ChEBI, there was a tool developed by an external group called Classifier. And what this tool does, it's a really clever tool, and it uses patterns of chemical structural sub-elements sort of together in rules in order to suggest classification for a molecule based on the chemical structure. And so this tool has something like 9,000 patterns in it that correspond to about 4,000 classes that it's able to predict. And at the time when it was released around 2016, it started to be used not for the fully curated part of ChEBI, but in order to bring molecules into the pre-assembly or ready for curation part of the ontology. And this made a big difference. It allowed us to sort of bulk load some additional resources or molecules that would otherwise not have been able to be bulk loaded. But it's limited in the sense that it's based on these rules. It uses strategy for classification because it's an external tool. It's not perfectly aligned to ChEBI's strategy of classification. And as a result, the classifications themselves are not the best that they could be, even for automated tools. So it's a great tool. It's already used, but this problem is not fully solved for even though it is in use. So I want to come back to this overarching framework of what we're sort of thinking about here. So the idea is, how do we take new metabolites that we, well, not new, but new for the knowledge resource that we are interested in, and put them in some way into our knowledge resource automatically, using the information that we have available to us and what we can automate in a way that can grow with the knowledge resource, can be updated without manual work, and so on. And this led us to try and explore whether machine learning would be able to solve this task of automatically putting a molecule into the right place in the ontology. So this seems like a sensible question or a sensible approach to try. And of course, we were not the first to think of using machine learning for doing classification into an ontology. There's quite a volume of work with other ontologies. And even with ChEBI, we had tried some variant of this classification automation a good decade earlier. At that time, we had not seen such good results. And what I will report now, we will show sort of we didn't see so good results when we tried it the first time. So first, I just walk you through what we did. So we tried to answer this question, can we use machine learning? And the important thing is that we want to learn from the existing ontology, how to extend the ontology, right? So we want to take what we have already and then train a model which is able to extend it. So as our first exploratory research step, we tried a whole army of different algorithms, as one does. And so there were a few of what's called classical machine learning algorithms, like logistic regression, random forest. This is for prediction of class membership. So it's a multi-class prediction problem. And one deep learning neural network, which at that time, the first one we tried was a LSTM, long short-term memory network. And it's important to note that the differences between the classical network and the deep network are more than just superficial, more than just which network, because we also gave them different input data formats. So to our classical machine learning family of algorithms, we gave them the chemical fingerprints, which are a fixed length kind of feature string, which is calculated from the chemical structure. But to the deep learning network, we gave it the chemical structure directly as smiles, this line notation representation of the chemical structure. So before we could start with the machine learning, the first thing we needed to do was to make some kind of training data set out of the ontology, which would have features which were friendly to the learning task. So because the ontology, this is a little bit of a messy illustration here. This is the visualization of the parents of caffeine. So you can see caffeine there. This is how caffeine is classified in the ontology. And at the top here, you have the root of the ontology, chemical entity. So under chemical entity, you have molecular entity, and then you have main group molecular entity, and so on and so on. And you get down to purine, alkaloid, methylxanthine, and so on, which are the near parents of caffeine. And this kind of diamond-shaped classification hierarchy is very characteristic of chemical classification. So you have one root of the ontology, and then you have your molecular entities which have just a few parents, maybe 2 or 3 parents, but then in between you have a kind of explosion of, this is because each chemical class represents a certain feature of the molecular structure, but every molecular structure has many features. So every molecule or metabolite has many possible classes that it belongs to, and this means that your problem for classification, these classes all share lots of members, and they're big, sort of overlapping classes, and finding the distinguishing features in a machine learning task is quite challenging. So this is what's known as a kind of extremely multi-class, polyhierarchical, and unbalanced problem. This is really challenging for machine learning. So what you see here is the number of classes that have X number of members in the ontology, right? So if you look for a class that has, for classes that have 10 members, then members here means molecules that have a structure. Then within the ontology, there are more than 2,500 of them. So if you look for classes that have 500 members, so 500 leaf node molecules with structures, there were only, I think, 300 or so of them. So the challenge was to find a good balance between having members you can learn from to predict class membership, but having an interesting number of classes to predict class membership. So we wanted to explore different ways that we could slice up the ontology to create a learning or training data set in order to train algorithms. So what we did was we took different numbers of classes corresponding to different members, different numbers of members per class in a kind of subsampling approach. And we did subsampling in order to generate a balanced training task. Now, actually, as it turns out, as we've sort of progressed in this, for some of the machine learning algorithms, the balance for the training task isn't the most dominant factor affecting performance. But at the time, we didn't know that, so we were just trying to have a good strategy. So what we did was we took the entire ontology and we sort of automatically processed it into class memberships, example members, which are molecules with structures, and different sets corresponding to different training membership sizes. And then we passed those to different algorithms. So we have here a many problem kind of evaluation where we have performance across all of the classes in the prediction set, and that prediction set might have been 25 classes or 500 classes. We picked different sizes in this space. And the number of members in the class could have been 25 or 500. Again, we picked different sizes. So number of classes to predict, number of members to learn from, and finally different algorithms. So overall, what we observed is, as you might expect, that the predictive performance, the average predictive performance, decreases with the size of the problem. It decreases both with the number of members. If you have more members, you have a more complicated learning problem or training problem, because then this is just a feature of the size of the way that the chemical ontology works. If you have more members per class, then your classes are going to be more overlapping, which makes a more difficult prediction task. And if you have more classes to predict, then you have a more difficult prediction task. So on average, performance decreases with size across all algorithms. But what you see here is that there's one approach for which this general trend is not as strong. And that is, of course, the deep neural network, that we started with all these other different approaches and one deep neural network. And then we saw that although there was a general trend for performance to decrease with size, this was not nearly as true for the deep neural network approach that we used. And in fact, this was one of our main conclusions, that in fact, when we had tried to, let's say 10 years before that, to use machine learning with the state-of-the-art machine learning approaches at that time to solve this problem, that maybe those strategies just did not have enough capability to distinguish the large-scale chemical ontology classes, which were only having these small nuances differentiating them and being quite overlapping. And in fact, we needed a deep neural network to solve this problem. So this was quite exciting. So here you see the results sort of on average broken down for two of the different problem sizes. So you see here the 100 classes with 100 members to learn from problem size. And here you see that actually the best performance for this problem size by the logistic regression. So a traditional kind of, it's not hugely more performant than the other approaches, but this is the best performance in that problem size. But here you see the 500 by 100, which is much more realistic as a problem size for the Bio-Ontology. And here you see that you're getting a massive outperformance of the other approaches with the deep neural network. So that was a kind of a clear signal out of our experiment, and started to suggest that maybe we could really use this technology to build an actual tool that could do this task. So that was really exciting. One interesting observation we found was that some of the molecules would get no class predictions by the deep neural network. So just to recap, our training data and our testing data were drawn from the Bio-Ontology, and all molecules have some place in the Bio-Ontology. There's no molecule for which there is no chemical class that's relevant, because the ontology starts at the broadest level of generality. So we should never have a situation where it's correct to predict no chemical class. So here you see the molecule-wise. So molecule-wise means we did the averaging of the F1 scores across the predictions for the same molecule. So you can have multiple class predictions for a molecule, and then we calculate the F1 score based on whether they are correct or not. So mostly, if the molecule prediction, class prediction was correct for one or any of the predictions that were made for it, then it was predicted for all of them, and that's why your average F1 score is 1 here. there were a few for which you had maybe 3 correct predictions and some others that were incorrect. And then you had some for which there were no correct predictions. And it turned out when we looked into these, this kind of worrying tail bulge over here, we found that these had indeed no prediction at all. And so obviously they had no correct prediction. So then we looked at the sort of comparison over the best performing classes per learning algorithm. So here what you're looking at is for the different problem sizes, for 3 of the different problem sizes. The problem size, 500 classes, 25 members per class. Here we see the best performance were the logistic regression, discriminant analysis. Random forests also did quite well. When you get to more classes and more members, we're dominated by the performance of the deep neural network, as I mentioned before. But again, what we see is that there's not a perfect overlap between which were the best performing classes and different algorithms. So what this means is that these different algorithms are potentially picking up different features better. Basically, this is the conclusion from this. And so it really suggests that maybe in the end, to solve the problem comprehensively will require a combination of different approaches. This was also quite interesting. Some more general observations, we found that the classical machine learning approaches as a whole, taken together, gave really bad performance for classes higher up in the ontology hierarchy. And this makes sense because these types of machine learning approaches perhaps have a lower dimensionality space in which they can separate the class membership, and the members of these classes, which are high up in the hierarchy, are very diverse. So it's a kind of broad groups of underlying molecules which are overlapping and picking out the unique features for these higher up groups was maybe something which this kind of approach was just not able to address. That's fine. But then we saw another kind of interesting, so our deep neural network, the LSTM, gave bad performance characteristically for molecules which had aromatic ring structures in them, or kind of complex groups of ring structures. And this was, again, a kind of, so this would be something for which in the fingerprints, perhaps it was easier to pick up aromaticity and so on, but somehow when the network was trying to learn aromaticity and related features from the SMILE string, the input representation of the molecular structure, it wasn't as able to learn that. So this, again, it's something which, it's a very interesting thing that we learned, but has to do with the relationship between the problem and then the technology and what the technology can offer. So the next thing we did was a comparison to the state-of-the-art classifier, being this tool for predicting membership based on chemical structures, which is widely used at the moment, and therefore we tried to compare. It's not so straightforward because it doesn't do exactly the same thing, and the ontology classification that it predicts is not exactly aligned with KEBI. So what we did in order to make this comparison possible was we identified a metric, which is the number of paths, connections, so classification relationships between the true parent and the predicted parent. And this metric we were able to use in both cases, our predictions and the classifier predictions. And what we saw is that classifier as a whole gives many more predictions for an input chemical structure. This makes sense because it's based on rules rather than having learned where it was actually classified. So the number of parents for each molecule in KEBI was below 5, on average. Our network predicts below 5 parents, but classifier gives on average 8 to 10 parents for predictions. And the path length metric that I mentioned, which allowed us to kind of evaluate. So just to have an idea, the training data as well had some path length, which was not always one because of the sampling approach. So we selected membership not only for immediate parents, but also for parents higher up in the hierarchy. So here's our training data distribution. This is as good as it could ever, any prediction approach could have done. And then you see the different distributions on this path length metric. And you see the take-home message is that the classifier has better and worse performance than the machine learning based approaches. So because it predicts multiple parents, some of them are closer, some of them are further away. So I skip over that. So our summary so far is that we were able to predict ontology class parents for a molecular structure. And this is really exciting because it would allow potentially an approach which allowed you to extend the ontology using the knowledge, using a model trained with the knowledge that's in the ontology, and then you could kind of grow in this way. There were some limitations. It would be good to have some sort of ensemble to allow the strengths of one approach to mitigate for the weaknesses of another approach. And we thought that explainability was a really key factor, which was not addressed with the deep neural network, which showed the best performance. So then we thought at that point, the transformer-based architectures, which have at least some form of intrinsic explainability. So our next experiments were to transform this problem again a little bit into a strategy which we could use with transformer networks. So just for anybody who doesn't know, transformers are the deep neural network architecture that rose to fame with the paper titled "Attention is All You Need," and they have this additional attention mechanism which gave them enormous power in natural language processing, and they quickly became the network architecture that was being used for everything and actually generating quite good performance. And another paradigm shift which was related was this possibility of not only training a network for the prediction task, but having this additional pre-training, which is kind of an unsupervised approach where you give the network a whole lot of examples of the data that you're trying to predict with and mask a part of the input data and get it to predict back the missing parts as a kind of a pre-training phase, right? So we wanted to use a transformer-based network. We wanted to add to what we had done before, a pre-training phase. So we selected pre-training a big data set from PubChem. We sort of randomly sampled it in order to make a pre-training data set. And then we used a transformer architecture with this extra step of having pre-training and predicted class membership with the same task as before. And what we saw was that the transformer-based network outperformed the LSTM-based network. This is perhaps expected because these transformer-based architectures have been outperforming other architectures for a lot of tasks. And we were able to quantify how much it outperformed. So for example, you see here the molecule-wise and class-wise F1 scores, and shifting towards the right is evidence of better performance. So here you have more classes that are getting closer to an average F1 of 1. Yeah, in the Roberta, which is a particular implementation of the transformer-based architecture. Right. So then our big objective was to see if we could get any interpretability out of this transformer-based network. So the attention weights in a transformer network are a relationship space from the tokens in the input to the tokens in the same input. So it's what's called self-attention. And attention is just a way of carrying information across a context so that you have that information at a later point or at a different point when you're processing another part of the input, and you can integrate a wider range of different inputs that way. So here, what we observed was that there were different patterns of attention in different layers in this network. So attention has multiple layers. And in some of the lower layers, the attention was always just looking right next to sort of what's the next atom, what's the previous atom. So really local attention, or mostly local attention. But then we observed that the higher layers of the attention network were showing patterns or clusters of attention which really corresponded to, so here you see this nitrogen and this aromatic double ring cycle. So this attention really corresponded here to the pattern within the molecular structure, which was the right part of the molecular structure that it should be looking at in order to make the ontology class prediction. So this was really super exciting for us, these patterns of attention and the way that they related to the ontology class prediction task. And this led us to wonder, to sort of hypothesize whether this ontology class prediction task might be more broadly useful. It's sort of a way that the network has had the opportunity to learn some exciting or interesting things from the ontology class prediction task, which may be useful for other prediction tasks. So in summary, so far, what we've seen is that it is possible to predict class membership. Transformers have the best performance, but still not perfect performance. Ensemble-based approaches balance the different strengths and weaknesses of various different approaches. Some form of interpretability is available through the attention weights of the transformer-based networks. So now I want to talk a little bit about ontologies and machine learning for discovery, for sort of getting the information from these ontologies, and this is the virtuous cycle part. So what we asked ourselves was whether this information that we had from the ontologies could actually improve machine learning for other tasks, right? So here we conceived of a kind of multi-layer process where we have, as is normal now, sort of separation between general pre-training, which gives the network the understanding of the input structure data type, and the prediction task, which is targeted to a specific problem. We envisioned the ontology training as a sort of intermediate step, which gave meaningful structures from the domain knowledge that was represented in the ontology to the network that we hypothesized might give the network the possibility to have a better generalizable understanding of the domain than it would have just from data alone. This was the hypothesis that we set out to test. And in order to test it, we chose toxicity prediction, because toxicity prediction is a hard challenge in cheminformatics machine learning, where you have many different strategies and a lot of data, so we could sort of compare our results to other results. And the design of our experiment was we used the data from the Tox21 challenge. This is a publicly available data set of toxicity against various different toxicity receptors, where you have a kind of pre-existing separation into training data and evaluation data, which other people have also used. So this was really nice that we had this, that we could also then try our approach on. And what we did was we made two different sort of sides of the experiment. One in which we did our, so both sides of the experiment got the same pre-training with the same pre-training data set that we had from PubChem. And then one of the sides of the experiment got this ontology pre-training extra step where before we tried to predict toxicity, we just gave the network the same task that we had developed before. We trained it to predict chemical ontology classes from Kebi. So pre-trained model from the standard pre-training either predicted the toxicity directly, or learned to predict toxicity using the training data from the TOX21 dataset, or went through ontology pre-training and then learned to do toxicity prediction. And what we saw, which was really great, was that the ontology pre-training side of this experiment had better performance, first of all, in absolute terms. Second of all, it arrived at that better performance with sort of quicker, so the training time was reduced. And as you can see more most clearly here, it also improved or seemed to buffer the network against too early overfitting. So you can see this kind of characteristic. So the blue is with ontology pre-training, the red is without. And you can see here this kind of characteristic decrease in performance that results from early overfitting, which you don't see in the blue line. And this shows that because this TOX21 dataset is quite a small dataset for a machine learning with a deep learning approach, and this is overfitting is a kind of typical feature of when you have too little data and you don't have a good data set to learn from. So we thought these results were really exciting. There's a preprint out, it's under review at the moment. I hope that it will be reviewed somewhat favorably, but we'll see. We also compared the absolute results to the state of the art. And here it's important to say that our model did outperform the state of the art for machine learning based approaches on this task without feature supplementation. But in fact, the best performing approaches for toxicity prediction do use feature supplementation. That means they use algorithms to calculate additional features based on the chemical structures and then put those additional features together with the structures into the machine learning pot. We didn't do this, and we only compared our performance with other models that also didn't do this. But it would be hypothesized that if we did this, it would further improve the result. We also observed that the pre-trained model had more clusters of sort of meaningful clusters in the attention weights than the not pre-trained model. So this shows that the pre-training has given the model or trained the model attention to be more meaningful somehow. As a kind of global metric for this meaningfulness, we calculated what's called the entropy of the, or how random the attention weights were. And we found that there was a systematic effect that with the pre-training, the entropy was lower, and lower entropy means more order. And you can see that here in these. The top line here and here is for with pre-training, and the bottom line is without pre-training. It's not that there's no order in the attentions for the system without pre-training. And we would expect that from the task itself, the network has to learn some meaningful ordered patterns, but the order is clearly more substantial if it's had the ontology pre-training compared to. So, in conclusion, what we found is that for this task in general related to metabolism, we can use the knowledge we already have in the ontology in order to create predictive models that suggest how to extend the ontology. So this has the potential to substantially enhance the knowledge capture and assembly and curation process, and this is really exciting. But in addition, this process of training a network to predict ontology classes is itself something which teaches the network the ontology. It teaches the ontology to the network, and this improves downstream predictive performance on other tasks. And this is has then potentially quite wide applicability. So yeah, we will be in the future. So this is this idea of a virtuous cycle between knowledge curation and discovery. And we worked for kind of accelerating that cycle using machine learning together with the ontology. As our next steps, we have a tool which does this ontology prediction task. It's not yet online, but it will be soon. We're going to do a user-facing study of those attention visualizations, just to sort of see, I mean, interpretability of machine learning is a kind of hard challenge, and we would like to see what expert chemists really make of how useful those visualizations are in a real user study. We are exploring other architectures to be able to use more axioms from the ontology as input knowledge. At the moment, the only axioms we use are the subclass axioms. And we're looking to test this approach out on prediction targets relating to metabolic health. So with that, I say thank you. I'll be open for questions. I just acknowledge my wonderful co-workers. Most of this work has been done by a team at the University of Magdeburg, especially Martin Glauer, who is a whiz with implementing neurosymbolic machine learning architectures in very, very short times, and this is really great. And the user study will be run by Mari Wozni, who is here in my group in St. Gallen. Thank you for listening.
Thank you so much, Janna. Fantastic talk. And you have a few questions on the chat. So first one, could the success of AlphaFold on protein structure be translated into a method for small molecules?
AlphaFold in protein structures and a method for small molecules. So if I understand correctly, a lot of the knowledge that goes into AlphaFold is based on sequence similarities between the between different organisms, and then that allows the model to sort of learn more relationships between sequence and structure across homology groups, and I think it's really a very exciting architecture. It would have some potential translations into the small molecule space, but I'm not sure if anybody is working on that. It's quite a big and complex architecture under AlphaFold. So, and in a way, we don't need to predict the same thing, because prediction of three-dimensional structures of small molecules is not so difficult, right? So I don't know if that answers the question.
Hopefully, yes. The next question is, when you say that machine learning have a hard time learning the ontology structure, is it because it's not necessarily hierarchical? Sorry. And could ontologies be transformed into a more hierarchical, or what would be the approach to tackle this?
So I think I said that traditional approaches to machine learning, which is like linear logistic regression and random forest and so on, had a hard time learning the classes to predict class membership for classes higher up in the hierarchy of the ontology. So the ontology does have a hierarchical structure. It's polyhierarchical and the classes overlap. It's not strictly hierarchical. If in the sense that you don't have pairwise disjointness between members on the same level, right? So if you had pairwise disjointness between members on the same level, I think that it would be an easier task to then predict membership. And then maybe it would have worked well enough with the older approaches. But in order to learn to make accurate predictions across this polyhierarchy that shares a significant portion of members, I think it doesn't work with the classical approaches. But they are still very good at predicting smaller classes that don't share members. Does that make sense?
It makes sense to me, at least. Hopefully you answer the question. Another question. So how does the ontology quality or the different ontology use affects machine learning performance? For example, if you use PubChem, which has an RDF model, how could it impact?
So we don't use the representation of the ontology in RDF or in OWL or whatever. We use the classification in some kind of abstract sense because we put the classification into the training data directly. So we pre-compute or pre-realize all the possible parents for a molecule and then use that to produce the prediction task. So the question is whether a different ontology which has different level of quality would affect the machine learning model one way or another way. I think we don't have any empirical data to answer that question, but I would hypothesize that the lower the quality of the ontology, the worse the performance of the model in prediction. And this we could do an experiment if we could get someone to agree to give us an ontology that they thought was having a lower quality. But I could imagine that quality evaluation is a little bit through the lens of the beholder. So we could turn it around. We could say, well, if it's really difficult to predict, maybe there's a problem with the classification, right? Maybe this could be turned into an empirical metric for quality, or not.
I have, and I think it's related to this one. Sorry. Do you think that ontologies, using ontologies in machine learning can produce some kind of bias?
Yes, yes. So it certainly will produce some kind of bias, right? And the first bias is that your ontology will improve your results only where your ontology has data. And where the prediction problem doesn't have a good representation in the classes of your ontology, you will not improve your prediction. So you're definitely adding bias. I think any kind of addition of knowledge or whatever has at least the potential to bias the prediction problem. And you have to evaluate whether this bias is worth it. Whether what you're adding from the ontology in the form of knowledge is going to be the right thing for your prediction problem.
Yeah. Especially in the chemical space, because it's so huge.
Yeah.
And our knowledge is so reduced.
Yeah. But I think the relevant challenge is not necessarily the size of the ontology, but how representative it is, right? So how broad. Yeah.
So you mean how does represent the chemical classes, right?
How broadly it represents chemical classes across the relevant possibilities of chemical classes. Yeah.
Makes completely sense. Thank you. More questions from the attendees. May have missed it, but do you use SMART's substructure matching to classify your molecules?
So the SMARTS is the approach that the classifier, the existing tool, the state-of-the-art, it uses SMARTS. And we do not. We use machine learning, and the model classifies the molecules. So there's no explicit rule. There's just a trained model which can do classification. SMARTS is a representation of an explicit pattern in a chemical structure that can be used or associated with a class.
Okay. And the last question in the channel, the panel until now, did you consider spectral attention within your transformer architecture? given its success in distinguishing subgraph structure, I'm curious if it might be helpful for addressing some of the challenges in learning second-order proximity within ontologies.
Spectral attention is not an expression that I know. I don't know exactly what that means.
I don't know if Tiffany, that is the person that is asking this, can speak out to clarify this. But I have some questions in the meantime. For example, in order to refine the Kebby ontology, have you think about using multimodal learning? Like mixing imaging with expression, with the structure?
So absolutely, I think this would absolutely be the same, right? So it's just for our approach, what you need is a link between the input data and some kind of ontology classification. And this link is what you then build your pre-training exercise out of. And it can just as well be 5 different data types, or different ontology classes and so on. So it's equally applicable to a multimodal context. Of course, you probably wouldn't, we use chemical structures, and I think the chemical structures are a kind of information-rich data type. Images are also a super information-rich input data type, but then there would be some data types that maybe you didn't need any kind of classification because they are what they are, right? Binary features and so on.
Okay. While waiting. Okay.
So I don't fully understand the question. I think about the attention, and I would be really happy to be to send me a link or some additional clarification, please send by email, and I will be really happy to look into and be informed by that.
Yeah, Tiffany says that. Perfect. I have some more questions if we have time. Regarding chirality, chemical chirality, it's very important for drug discovery.
Yeah.
And how is this handled in Kebby?
So Kebby distinguishes stereoisomers, and so this may be in the input data as separate stereoisomers may be represented. They are represented separately. I think you can tell from the smiles, because if it's needed, the smiles will include a representation of chirality around a certain bond. So I think it's there in the training data set. We didn't specifically evaluate this as an aspect for good performance or bad performance. We did find certain chemical features characteristically gave poorer performance. I mentioned aromaticity. Radicals were another one that were not. But the question whether the classes will, I think for the prediction task, what's important is what's captured in the classification. And I think the classes, there's not so many classes where stereochemistry is part of the class definition, right? It's relevant at the molecule level, not as much. There are a few, but not so many. Anyway, something to explore maybe.
Yeah, that was my next question. And also another question regarding metabolites. Some metabolites can be used as drugs. And how this is represented in Kebby?
So Kebi has this other classification, which is about how the molecules are used or whether they are metabolites. And this is the role, it's called role classification. In this study so far, we did not use the role classification. We do plan to. It's kind of one of our next steps is to feed in the role classification as well. But so far, we only use the structural classification, and the structural classification is agnostic about how the molecules are used.
Right. And my last question would be, if have you tried graph neural networks
Yeah, we did try them. So this was one of our sort of experiments somewhere along the way. We tried, so between the LSTM and the eventual architecture, which we're using now, which is based on transformers, we also tried graph neural networks, and we did not find them doing better. So they did not outperform the just using SMILES and a transformer-based network. And this was a surprise to us at the time because we expected that the graph neural network would perform better. It's still a little bit an open question whether maybe some other variant of the architecture could have done better, but they were also slower, so slower to train.
Yeah. And have you tried zero-shot learning for prediction of the classes for classification?
So no. So I think our setup wouldn't support zero-shot learning because the class label in the prediction that it has to predict the class label, it does not associate it with any additional semantics. So there's nothing that would, if we took a class that the model had not been trained on, there would be no information to the classes that had been trained, right? We would need to also feed in the kind of class information as a separate step, maybe with a, knowledge embedding strategy.
Perfect. I don't have any more questions. I don't know if there is another one. Let me check.
I have a question before. While you're checking. Thank you so much, Janna. That was a great talk. I'm curious about the tool you mentioned that you'll be releasing. And I was wondering if when you are giving a new or, chemical to classify, do you specify at which level of the hierarchy you would like it to be? Because I assume if it just gives back chemical entity every time, that wouldn't be very helpful.
Exactly.
At all.
So,
and since there are so many hierarchies in there.
So the tool will, so the way that we designed the model is that it predicts all the relevant classes that it finds membership. So it will always predict chemical entity as well as the other ones. But then we use the normal kind of logical hierarchy to try to figure out the best classes. So lower in the hierarchy is ranked better at this step. So we always try to find what are the lowest parent classes, the closest to the leaves, the more specific, and try and return those.
Okay, that makes sense. Thank you.